Today, we begin a new season of the Polar Seminar Andrés Barbosa, the first of 3 sessions before the summer. It is my great pleasure to introduce today to Beatriz Fernández-Marín, a young but very expert researcher with an impressive track record in polar research. So she got her PhD in the University of Basque country in 2012. Later, he was in University of Innsbruck with a Marie Curie individual fellowship. Later, she came back to Spain with Juan de la Cierva, and later she got a Ramón y Cajal grant. So at this moment, in between the Juan de la Cierva and Ramón y Cajal, she started her own line of research about cross-tolerance to desiccation and freezing. Actually, is with the Ramón y Cajal in the Basque Country University. So now is your time.
So thank you, Asom, for the nice introduction, and also for the opportunity of being here today, sharing some of my data. I'm plant ecophysiologist, so my area of expertise is related to photosynthetic organisms and how they deal with environmental conditions. I'm specialized normally in abiotic factors. For today's presentation, what I want to talk to you about is about terrestrial photosynthesis at the poles and some challenges and peculiarities that these organisms show. So the structure of my talk is based on 3 main blocks. I will give you an overall introduction first, and then I will show you 2 different blocks of results. So within each of these set of results, I will give you some context too. For the introduction, my intention is to explain some of the key concepts of my talk, and all of them are included in the title. So I will start with the essential one, with photosynthesis. Photosynthesis is a fascinating process which consists essentially in the fixation of carbon from the atmosphere into sugars. And this process happens in the presence of liquid water and is mediated by the energy of light, of sunlight. As byproduct, oxygen is produced, and this is what we breathe, for instance. If we look a bit more into detail in a plant cell, in a photosynthetic plant cell, photosynthesis takes place in a specific compartment, which is the chloroplast. And there we can distinguish 2 sets of connected reactions. The first set of reactions is basically a conversion of energy from light to chemical energy. So essentially, in this step, we will pass from having photons, light energy, and we will end having electrons moving from one molecule to other in oxidation and reduction reactions. This process is physicochemical, so not real enzymes involved that much. The second set of the reactions consists on the assimilation of carbon from the air into biological molecules, into sugars. And this process depends strongly on enzymatic activity. Today in the talk, I will not talk that much about CO2, not about oxygen, but I will focus on 2 of the essential elements of the photochemical part of photosynthesis, which are light and water. The next concept, the poles. The official definition of what the Arctic and Antarctic polar circles are is essentially this. Within these land areas, at least 1 day per year, the sun will be above the horizon for 24 hours. This happens at latitudes above 66 degrees, approximately. And this is why, because of the angle of inclination of the rotation axis of our planet. However, I must confess the definition of pole that I will use in my talk is not exactly that. I have broadened by myself the definition. And I have gone down to 60 degrees in latitude. And this is why, because of the physiological point of view, talking about plants. And whenever the sun is up to 6 degrees below the horizon, we do have an effect which is called midnight twilight. That means that we don't have direct sunlight incident over the plants, but we will have diffuse light. So the sky will not become black, but there is some light. And for instance, we could be able to read a book with this light. It's a very dim light, but it's still light. So from the physiological point of view of a plant, the true definition of the poles will be this one, 60 degrees. Next concept. What are the challenges that a living organism has to face when living at these high latitudes? Well, I will start showing you some examples of the challenges we as scientists have to deal with when working there. More specifically, this is my hand working during a summer campaign in the Arctic. Are you able to tell me how many mosquitoes are already in my hand? There are 4. So only in one hand. Imagine in the rest of the body. They are very clever and they can find us even if we're trying to protect ourselves. Luckily for photosynthesis, mosquitoes are not challenging at all. So we will focus more on the elements that have to do directly with photosynthesis process. As I told you already, I will not talk that much about CO2 because even when there are some oscillations on the concentration around the plant along the day or along the year, which is a time scale relevant for a living organism, we could say that the CO2 concentration is pretty stable and in any case is not limiting for photosynthesis. However, what happened with the 2 other main elements, with light and with water, liquid water? Well, in terms of 1 year or even in terms of several hours within a single day, any of these 2 elements can change several orders of magnitude. So plants living there, photosynthetic organisms living there, must be adapted to these changes in availability for 2 elements that are absolutely essential. And talking about light specifically, there are changes on the intensity, but there are also changes on the time this light is available. We call this photoperiod. How many hours of light do we have per day? This is photoperiod. And at high latitude, photoperiod can be extremely long, even 24 hours of continuous light, and even several days consecutively under these conditions. This is not an easy situation, even for us. We have problems to fall asleep when we are working in these areas or people living there, so we need to darken our rooms or our eyes. And actually, many of our physiological processes in our body are controlled by what is called a kind of biological clock, which is the circadian rhythm. Alterations on these rhythms between day and night, which is light is one of the regulators of this clock, can produce serious issues to our bodies. So, for instance, proper darkness, proper rest enable us to repair and to restore some of our molecules. And this is a study from this year. The influence of too long photoperiod in our body has long-term damaging effects. So it's related, for instance, with the increase in the risk of several metabolic diseases. What happened to plants or to photosynthetic organisms? Actually, something similar. They also have many different physiological processes that are regulated by this circadian clock, and they need to have some hours of light or of darkness in order to have a good equilibrium in this metabolism. So very long photoperiod can be also damaging for many species, and different processes related to development, to phenology, or even directly to photosynthesis can be affected. For some species, staying under continuous light is even lethal. So not all species can be adapted to this strange condition. What happened with liquid water at high latitudes? Well, in essence, the Arctic and Antarctica are virtually deserts. So precipitation on average, there are heterogeneities, of course, but on average are very low both in the Arctic or in Antarctica. So the availability of liquid water coming from precipitation is very, very low. In addition, of course, these are two regions really, really cold. And this will induce other effects, negative effects on photosynthetic organisms too. So whenever we are at sub-zero temperatures, water can become frozen. And having light plus freezing temperatures is very risky for a photosynthetic organism. Why? Because I told you before, this set of reactions in the photosynthesis depends on enzymatic activity. So whenever the tissue becomes frozen, enzymatic reactions are not possible anymore. However, the absorption of light energy by chlorophyll, which is a kind of physicochemical process, will persist. And this energy absorbed by chlorophyll will necessarily be released. And in a situation where no photosynthesis, no fixation of carbon is possible, this excess of energy will oxidize other molecules and can end in the death of the cell or even of the plant. So this is a high photooxidative risk, low temperature plus light. In addition, low temperature, if this induces the formation of ice within the tissue, will indirectly produce the dehydration of the cells. This is because water will first crystallize outside the cell in the apoplast, and then subsequently will grow up, taking water from the inside of the cell. In fact, from a physiological point of view, as a plant being exposed to a strong desiccation or to freezing share many physiological consequences. In the same way, the protective mechanisms are also very similar. So the last question for the introduction would be, under these conditions and with these challenges, which photosynthetic organisms is able to live there? And the answer is actually many, and many different. Here is a simplified phylogenetic tree of the green lineage of photosynthetic organisms. And I will show you now, we can find many different taxa growing either in the Arctic or in the Antarctica. Vascular plants, there are vascular plants at both regions. More than 2,000 species in the Arctic, only 2 in Antarctica, but there are. Bryophytes, so you know, mosses and liverworts. We can have almost 1,000 different species in the Arctic and a bit more than 100 species in Antarctica. And green algae, which is a non-very specific name we use to refer to actually a polyphyletic group of different organisms. Both of them, the streptophyte and chlorophyte green algae, can be found in both regions. So as a conclusion, I mean, the number of species is lower in Antarctica, but the diversity of different types of photosynthetic organisms is quite high at both sides. So we move now to the block of the results. And before that, I have to share with you that, of course, these data are not taken by myself alone, but in cooperation with my current group in the University of the Basque Country, and with my former group in the University of La Laguna in Tenerife, and of course, in cooperation with several other colleagues from different institutions. Also, this cannot be done without funding, so these results have been receiving funding from different institutions, such as the European Union, Spanish Ministry of Science, or BBVA Foundation. So let's start with the first block of results. I will focus now on one of the elements, on light. A chloroplast has to be ready to change from a harvesting light mode, so a photosynthetically active mode, into a dissipative mode. This means there is a situation when the light energy is more than needed for photosynthesis, so this excess of energy must be dissipated as heat before it can oxidize molecules in a risky way. So within the processes inside the chloroplast to change from the harvesting to the dissipating mode, this, what is called the xanthophyll cycle, is one of the most important mechanisms. Essentially consists of the interconversion of 3 carotenoids through reversible reactions. And we have 2 ways of measuring or estimating how photoprotected a chloroplast will be. We can quantify the total amount of these 3 carotenoids, which we call the total xanthophyll cycle pool or the total bath pool. And this will resemble, in a parallelism, to quantify how many umbrellas potentially useful for this chloroplast are there. After having done a very deep survey of literature covering more than 800 species, we have concluded that for a non-stressed chloroplast, this value, the total amount of xanthophyll cycles per chlorophyll, is approximately 50 millimole per mole. So above this threshold, we can say that this chloroplast is under a very photoprotective demanding condition. The second way of estimating how photoprotective the chloroplast is to study the proportion of those carotenoids that are directly involved in the dissipation of energy, which are androxanthin and zeaxanthin. Continue with the parallelism, will represent how many, which is the proportion of this total amount of umbrellas that at a specific moment are open, so are working in the dissipating mode. Again, after studying many different species from all around the world, not only in the poles, so from all latitudes, we concluded that approximately 0.2 is the proportion of a control plant. This value or lower represents a chloroplast which is not under high photoprotective demand. So, in a low latitude with normal day-night conditions, this will be the evolution of this proportion, of this deepoxidation index. We will have very low values before sun rises. This will increase. We will have the maximum around midday, and then this one will decrease again in the afternoon. This was the classical view of how the xanthophyll cycle operates, in principle mediated by light. But last research, and we had made some contributions to this current view of the xanthophyll cycle now, has revealed that not only light, but other stressful conditions, such as freezing or desiccation, can also induce an increase on these photoprotective molecules. And this is why, because these molecules are directly involved in the dissipation, but are also, this is something, are also involved in the structural preservation of the membrane integrity within the chloroplast. So they have a dual role, photoprotecting from excess of energy and photoprotecting from destabilization of the membranes. So our question at that point was, how does the xanthophyll cycle operate, work, under high latitude conditions? And to answer this, we have been working in different field campaigns in different locations. I will show you now. This is heat area in Finland. So this is a taiga. Still, we do have several trees. This is farther northern in Finland, Barrio Research Station. Very few trees. We are already in the transition through the tundra. We were also working in Antarctica, no trees anymore, and also in the tundra in the Arctic in Spitsbergen Island in Svalbard. So our first result is related with the amount of the total battery of protective compounds. How big is the size of the xanthophyll cycle pool at high latitudes? This is the average of all the species we have been working on with, and our conclusion is that for those species living in the tundra, the total amount of the xanthophyll cycle pigments is significantly higher than for the average. So first, a small conclusion related to xanthophyll cycle at high latitudes. The pool of this protective group of compounds is higher than at lower latitudes. Next question was, how does this cycle work at high latitudes, but if we have a normal photoperiod, which means more or less 12 hours of light and 12 hours of darkness? And to do that in high latitudes, we can take advantage of 2 days per year, the equinoxes. So at that days, we do have naturally more or less 12-hour photoperiod. So we were working in Antarctica at the end of March in the spring equinox, comparing 2 species, one vascular plant and one moss. We were monitoring how the xanthophyll cycle, the epoxidation index, oscillates. And at that period, we already have true nights, so are highlighted here with dark color. We can see how the irradiance of the sun was changing, so peaking maximum values at midday. And in red, we have the temperature. So what we obtained in this experiment was essentially 2 types of results. First of all, the operation of the cycle apparently was normal, so the maximum peaks were at midday and the minimum at the end of the night. Second, if we pay attention to the temperature, the second night of the experiment, we did have freezing temperatures. If we pay attention to the deoxidation state, it didn't increase due to these freezing temperatures. So our 2 conclusions here are that the cycle operates regularly if the photoperiod is of 12 hours, and that the cycle has low sensitivity to freezing temperatures in these latitudes for this species. Next step was studying them under a natural condition of truly long photoperiod, so 24 hours of continuous light. These conditions we find in June, July in Svalbard Island, and we were doing something similar, monitoring the xanthophyll cycle operation along several days. If you see here the oscillation of the sunlight, the radiation, you can appreciate that we started with high values because they were some sunny days, but thereafter the maximum values were low because we have cloudy days. What happened to the xanthophyll cycle? In this case, we didn't get a normal operation, so the maximums were not always at midday, but we were able to appreciate a decreasing general trend that correlated better with the cumulative radiation. So the cloudiest the day, the lower the values we got in this index. Then our conclusion here is that under 24 hours of continuous light, the cycle is not really operating as we have known before. And the next study, we were also working under almost 24 hours of natural photoperiod. And we tried to go a bit more in deep into this fact of how does the cycle working if we have too many hours of light. So again, we had day and night, so the radiation is changing, but during the night we don't have total darkness. And we couldn't find the normal operation of the cycle. In fact, one of the maximum values was reached during the nighttime of the second day. And what happened during this nighttime? We have a cloudy day the day before, but during this nighttime period, the sun directly arrived to our plant, so we had the effect of a sunbeam direct to our species. This was not very long, 15 minutes, and was not very intense because of the angle of the sun in the sky is very low. So we arrived to 150 micromoles low light. At midday, we can be at 1,000 something, so 1 order of magnitude higher. This dim light, and for a short period, was enough, on the one hand, to raise the total amount of the xanthophysical pigments, and on the other hand, to raise the deepoxidation index. So our conclusions here were that the response was related to light and not directly to the time of the day, although it is also important when the light appears. So we have an exacerbated responsivity of the cycle if the sun was incident on the plants during the night. So we move now to the second and last block of results, and we change the element. I will be talking now about water, or more specifically, about the lack of water. So we try to understand how these organisms do to face very low availability of liquid water. This can happen because of low rain, but also because of low temperature. So whenever the water is frozen, it's not available for photosynthesis anymore. And most of the results I will show you now are located in Antarctica. So the flora there is dominated by bryophytes and lichens. And these two groups of organisms share a very relevant physiological property. They are able to desiccate and rehydrate, and they are unable to keep water inside their tissues for too long time. It depends, of course, on the species and on the size of the organism and in the anatomy, but compared to vascular plants, they are not able to keep water inside that long. So as a consequence, most of the species of lichens and bryophytes are tolerant to desiccation. So either because there is no liquid water and they desiccate, you can see how the cells shrink in the absence of water, or because of the low temperatures, when the cells become frozen, those can change a lot in volume. They become desiccated in both cases. And what happens to a cell when it's very, very dry? So below approximately this water contents, 0.1 gram of water per gram of dry matter of the tissue. The content, the cytoplasms, the organelles, will enter in a specific state which is called the glassy state. By comparison, in a crystal, we do have an organized network of molecules, and the properties of a crystal are the rigidity, so it's very difficult to move one molecule against other. In a glassy state, we do have the disorder that is typical from a liquid, but molecules are really immobilized. So what is physiologically relevant about this state is that the cell content is more or less stable, enzymatic reactions are virtually impossible, and deterioration reactions are really slowed down. The properties of a glass, so how rigid will be this material, depends on the composition of the matrix, on the proportion of water and solutes, and also on temperature. And we can estimate how rigid a material will be by measuring a parameter which is called glass transition temperature. So in essence, the higher the glass transition temperature is, the more rigid, more stable the material will be. And we have examples of this in our kitchen at home. So what we do to preserve food for longer, we play with this ratio. So we decrease the water-solute ratio by either increasing the concentration of solutes, for instance, adding sugar or salt, or we remove the water, for instance, drying food. The other option we have is to decrease a lot the temperature. In any of these situations, molecular mobility is very low, and also these degradation processes are low. So, to give you a number, what is the normal transition, glass transition temperature for a plant who lives in low latitudes? This one, for instance, is living in the Pyrenees. So more or less, plus 30 to plus 40 is the range where the glass transition temperature is or has been measured for low latitude plants. What happened when we study this parameter in Antarctic species? In this case, we compared a red intertidal macroalga, Pyropia, which is really tolerant to freezing and to desiccation, with a relative, also a red alga, but from temperate regions. The glass transition temperature of our temperate algae was more or less as our temperate plants, around 40. But the glass transition of Pyropia was much higher, almost 50 degrees centigrade. We have been measuring other organisms, such as lichens and terrestrial algae in Antarctica. And when comparing these two, Mastodia, the lichen, is much more tolerant to freezing and desiccation. What did we obtain? Again, in both species, the values are higher than this threshold of 40 degrees. And in the more tolerant species, we got higher degrees, 55. Now, in the last Antarctic campaign, these are data from spring, and still we are processing them, we have been measuring glass transition temperatures on different species of lichens and of bryophytes, and these are our preliminary results. Most of them, again, are above the threshold of 40 degrees, and only one species of moss, which is this one, Brachytesium, showed lower values. So our first conclusion related to water, it seems to be that most of Antarctic species have very high glass transition temperature when in the dry state. Second question is, let's think of the long-lasting viability of these organisms. When they are frozen or desiccated, which physiologically is similar, how long could be they alive? So in this study, led by my colleague Alicia Pereira Castro, we were studying different species of bryophytes. We kept them dry for 1 month and studied by photochemical activity, which means is the potentiality to make photosynthesis, if they were alive or not after 1 month in the dry state. What was our result? As you see, some of the species decreased dramatically, the viability, while others remain more or less viable. So the longevity in the dry state seems to be species dependent. So what we have done during this last campaign is to deepen a bit more in this issue. and try to perform a longer drying, but a bit more similar to nature, so with a slightly higher relative humidity. And the experiment was started in March, so I'm very sorry because the time point 6 months we will take next week, so I couldn't bring for you the final result of 6 months. I will hope I can tell you later. But I already brought data from 3 months, which is moderately long period. And we have been comparing bryophytes and lichens. This is our result so far. Under these conditions, most of the species were able to keep viability quite well, but one of the species already showed a decrease. And which is this species? Brachytetium. Do you remember this species? I saw you before in a different slide. This was the one showing the lowest glass transition temperature. So our next conclusion is that it seems in this Antarctic species the longevity is related to how stable they are in the glassy state. So in principle, higher glass transition temperatures will be connected to longer viability. And the last set of results also from this last campaign, we have been studying together what happened with water and with light in nature. So we started an experiment similar to the ones I showed you before, where we measured the xanthophyll cycle oscillations. In this case, we had some hours of darkness during the nighttime because it was March, the end of spring. And we started the experiment after a group of sunny days. So we had already some sunny, dry days. But we ended up with a cloudy and rainy day. So we have the transition from dry to wet during the course of the experiment. As you see here in green is the irradiance, 3 sunny days. The dashed line represents our experiment. And the blue bars represent cumulative rain of the day. So in the middle of our experiment, we had an event of rain. We compare one vascular plant with one moss, and I will focus on what happened to the moss. So as you see, when we started the experiment, the water content was around 0.1 gram of water per gram of dry matter, so close to this glass transition condition. Then we had the rain, and the water content of the moss, which is represented in dark green, increased. Immediately and closely related to that, the photochemical activity, so the potentiality to make photosynthesis, also rose up. And what happened to the xanthophyll cycle, the epoxidation index? We started with pretty high values that didn't change during the nighttime, so we were above 0.5. And then they decreased independently on the dark or light conditions, but because of the hydration. So we started with a bryophyte that was dry with high levels of photoprotective compounds, and when it becomes hydrated, these compounds decreased. So our conclusion here is that both light and water availability interplay in the operation of the xanthophyll cycle for these poikilohydric organisms. Now, to give you up with a kind of conclusion, I need to mention the only last concept which is in the title and that I haven't mentioned before yet, which are the peculiarities. So I hope I have already provided you some of the peculiarities of the Arctic and Antarctic photosynthetic organisms, and we could summarize those in 2 statements. To be able to make photosynthesis and to face the challenges typical at this high latitude, you must be efficient and dynamic in photoprotection, and you must have a very stable glass state in the dry state. So we have been studying this through the xanthophyll cycle, and and the longevity in the dry state through the glass transition temperature as parameters. So finally, I would like to thank once more to the team and collaborators, because this work is part of all of them, and also you for your attention.
Thanks, Beatriz, for your interesting talk and also for staying the time. So this is good. So now we have time for questions.
Hello.
Hola Beatriz, ¿qué tal? Enhorabuena por la charla, me ha gustado un montón. Perdón por haber llegado tarde, que nos hemos retrasado un poquito.
Perdona.
Me ha parecido súper interesante y súper chula todo lo que estáis haciendo y te quería preguntar, bueno, en general, igual hay excepciones, pero en la Antártida las tasas de fotosíntesis de líquenes y musgos al menos son bajas comparadas con la misma especie en otra latitud o con especies similares, ¿no? En principio hay ese concepto ahí más o menos asimilado, diría yo. Te quería preguntar si los resultados que tenéis están detrás de eso, parcial o no sé si totalmente incluso con esa actividad del ciclo Basque y tal vez otros fenómenos de fotoinhibición posibles y la relación que has comentado de la temperatura, la desecación, la luz y todo esto.
Sí, muy interesante tu pregunta. Nosotros también hemos medido fotosíntesis, o sea, la asimilación de CO2 como tal, que no he traído hoy aquí. Y te respondería la respuesta más fácil de todas, que es depende. Pero sí, en general lo que vemos es que en las condiciones en las que viven allí los organismos fotosintéticos en la Antártida, en general sus tasas de fotosíntesis son muy bajas, o sea que tienen la potencial capacidad de crecer muy despacio. Para varias de las especies, sin embargo, cuando estamos haciendo otro tipo de tratamientos y tenemos las mismas especies expuestas a temperaturas más altas, muchas de las especies, no todas, pero muchas de las especies tienen la potencialidad de hacer tasas de fotosíntesis mucho más altas simplemente por la temperatura. Entonces, un poco la conclusión sería, muchas de las especies que viven en Antártida no hacen más fotosíntesis porque no hay temperatura lo suficientemente elevada de momento, pero sí que es verdad que hay unas pocas que están, digamos, adaptadas a esas bajas temperaturas y no son capaces de incrementar las tasas de fotosíntesis incluso a temperaturas más altas.
Por la segunda que está relacionada con eso. Entonces, según lo que comentáis, si quitamos la competencia biótica, el incremento de temperaturas no debería tener un efecto significativo, dañino o sea, por ejemplo, en invertebrados pasa algo parecido, ¿no? Que les pones a altas temperaturas y se mueren a 30-40 grados, cosa que nunca van a experimentar con un cambio climático, nos morimos nosotros antes que ellos. ¿Es así?
Bueno, allí, para la mayoría de las especies fotosintéticas que hemos medido en Antártida, un incremento de temperatura, digamos que aceleraría, o sea, incrementaría sus tasas de fotosíntesis para la mayoría. Esto lo hemos visto sobre todo para los briófitos. Para el caso de los líquenes depende. Su óptimo de fotosíntesis es 15, 20, 25 grados, que en algunas ocasiones sí lo ven allí porque cuando hay sol sus talos se calientan por encima de la temperatura del aire. Y para algunas especies pues sería fatídico. Por ejemplo, Imantormia está adaptado a temperaturas muy bajas y el truco, el kit de la respuesta está en la relación entre respiración y asimilación de CO2, fotosíntesis. Entonces, como ambos incrementarán si incrementamos la temperatura, pues son los factores que regulan uno u otro los que tendrán la clave para saber si esas especies tendrán éxito o no.
Vale. Entonces sería más una cuestión de competencia biótica, ¿no? Con especies que llegan y que puedan hacerlo mejor y no tanto letalidad, ¿no? Que salvo el caso último que has comentado, raro sería el caso, ¿no? de mortalidad.
Sí, o sea, con los datos que nosotros, con las especies que hemos trabajado hasta ahora, son más las que, digamos, se benefician de un incremento de la temperatura que las que se perjudican.
Vale, y la segunda pregunta también muy sencilla. Hablas de la importancia de la luz. Entonces, ¿qué se sabe de los cambios en la nubosidad? Porque, claro, nosotros los biogeógrafos modelamos con temperatura y precipitación, incrementos de temperatura, y puede haber ahí un offset, ¿no? De que, bueno, el incremento de temperatura no sea tan bueno porque hay más nubosidad, ¿o cómo relacionaría? ¿Habría que meter en los modelos la nubosidad?
Sí, la nubosidad, y más que eso, pues la disponibilidad de agua líquida para los organismos fotosintéticos. Yo, por lo que he leído de la gente que trabaja de verdad en meteorología, que yo no trabajo, pues las predicciones son muy inciertas porque hay muy pocos datos tomados meteorológicamente allí, en la Antártida. Y lo que sí parece apuntar en general es que van a cambiar los patrones. Entonces, eso implica que puede cambiar el cuándo va a precipitar y el cómo, si es en forma de lluvia o de nieve. Y también la intensidad, o sea, si los eventos van a ser prolongados a lo largo del tiempo o intensos. Entonces, con todos esos factores en marcha, pues hay algunos de estos organismos que se benefician más de tener una lluvia fina, otros de aprovechar la nieve que se va derritiendo poco a poco, y otros pueden tomar humedad del aire cuando esta es muy elevada. Entonces, sí, habría que meter más factores en los modelos.
Bueno, claro, a escalas muy macro ya no es tan fácil. A escala local, en la base Juan Carlos puedes medirlo, pero si quieres tenerlo espacializado para todo el continente, pues ya es un poco más complicado, aunque se puede sacar índices de humedad y tal, pero bueno.
Pero bueno, sí, aquí en Antártida por lo menos distinguiría entre continental y Antártida marítima, que tiene una influencia así más directa de nubosidad, pues sobre todo en esa región sí sería importante.
¿Hay más preguntas?
Hola, Beatriz, ¿qué tal? No, en la línea que te estaba contando, Luis, es decir, nosotros hemos estado con parcelas de pisoteo en 8 comunidades diferentes durante 8 o 10 años. Todas se han ido recuperando a distintos ritmos, menos una, de líquenes. Entonces sería bueno ver si los ritmos de crecimiento son diferentes o si verdaderamente, es decir, esta que ha ido deteriorándose cada vez más cada año, es debido más a deterioros de condiciones ambientales del suelo o porque el crecimiento no han conseguido superar ese impacto de pisoteo que nosotros le hemos sometido. Luego, si quieres, ya lo planeamos, porque puede ser muy interesante.
Sí, desde luego ahí es clave qué especie está, pero sí, que sería interesante.
¿Más preguntas? Sí.
Bueno, la puedo preguntar desde aquí mismo. En relación también un poco a lo que estaba diciendo Javier, y en lo que has contado, en todas estas respuestas, además de estas adaptaciones fisiológicas, ¿crees que tiene influencia también características anatómicas de los propios, por ejemplo, en los casos de los líquenes, pero también en los musgos, tiene también que ver en estas respuestas?
Desde luego, sí, muchísimas. Solo centrándonos en los dos factores en los que me he centrado, la luz y el agua, simplemente cómo se forma la comunidad. Por ejemplo, en briófitos, pues algunos tienen una forma de crecimiento más en almohadilla o más en alfombra. En líquenes, algunos crecen más pegados al sustrato, otros se elevan en 3 dimensiones, son ramificados o no. Pues ambos factores, la luz y el agua, la van a recibir diferente, y en el caso del agua va a permanecer dentro de sus tejidos tiempos diferentes. Entonces, claro, todos esos factores van a hacer que, por ejemplo, soporten mejor o peor un periodo sin agua o de mucha luz. Y, por supuesto, cómo es la anatomía y la estructura y la morfología del individuo, pues va a tener un papel importante.
Claro, y supongo también que las sustancias liquénicas que puedan también tener unos líquenes otros también pueden jugar ahí, ¿no?
Lo mismo, pues o sea, hay unas sustancias que, por ejemplo, puedan tener unas características más hidrofóbicas o hidrofílicas, y algunas que son coloreadas o no. En ese caso, pues fotoprotegerían mejor o peor de la luz, o bien participarían en la absorción y mantenimiento del agua dentro del tejido.
Gracias. Una pregunta conectando con el medio geológico. ¿En qué medida el sustrato, litológico, la pedregosidad del terreno, la retención de agua subterránea o transmisión, crees que puede influir? Y conectando con esto, otra cosa adicional. Estas temperaturas que medís van unidas en la misma estación micro con la que medís otros parámetros y son muy locales, o utilizáis datos, por ejemplo, en las bases más meteorológicas generales.
Vale, pues por partes, el sustrato afecta, sí, por supuesto, en primera instancia, definiendo qué especies pueden aparecer o no ahí, porque tanto en briófitos como en líquenes, pues algunas son especialistas en sustratos rocosos, otras en suelo, y también depende de la composición, el tipo de roca. Entonces, primero, estarán o no, depende del sustrato. Y después, también las propiedades físicas del sustrato, pues si una roca se ha calentado durante el día, pues depende del tamaño y de las propiedades, permanecerá más caliente durante más horas durante la noche, por ejemplo, o retendrá más o menos agua en función de la porosidad, etc. Así que sí, definitivamente. Nosotros trabajamos más con la especie, no tanto con el sustrato, pero bueno, a veces sí medimos pues, temperatura, sobre todo temperatura y disponibilidad de agua del suelo, sí que tenemos medidas también, y radiación. Y luego, respecto a la escala, en general los datos que he mostrado hoy se refieren a estaciones meteorológicas de la zona, o sea que estamos más o menos en medio kilómetro a la redonda, es donde están las estaciones meteorológicas, pero de manera adicional sí que solemos tener microsensores para estudiar a veces la población o incluso el individuo exacto, porque la microescala es importante. Simplemente pues, que haya una roca o un obstáculo cerca hará que el momento y la duración, por ejemplo, de la exposición a la luz sea diferente. Entonces, sí, combinamos los dos.
Otra pregunta. Yo tengo una pequeña pregunta. Es que tú has mostrado como cambios muy directos, fisiológicos, ¿no? A los cambios de luz, etcétera. Y yo quería saber si en estos organismos que tú estudias existe un ritmo circadiano marcado genéticamente que se pueda expresar durante el día y la noche, o esto todavía no se ha investigado.
Para organismos poiquilohídricos hay muy poca investigación hecha. Nosotros tenemos algún trabajo, pero para ellos una parte muy importante viene marcada por el momento de hidratación y de deshidratación. Entonces, me arriesgaría a decir que para estos es más importante eso, el ritmo que marque la disponibilidad del agua, que no tanto un ritmo genético interno asociado a la luz. Y para plantas vasculares sí hay algunos trabajos, eso lo trabajan también los especialistas en molecular, y hay algunos estudios con especies del Ártico y también con especies cultivadas, que muchas de ellas pues no soportarían bien un fotoperiodo largo. Entonces sí que hay una componente genética, sobre todo en las plantas vasculares.
Genial. ¿Alguna pregunta más? Sí, otra. Las podemos hacer luego también en el kiosco, por si acaso, ¿eh?
No, yo dos preguntillas cortas. Has puesto unas cuantas especies, entonces ahí me ha entrado la curiosidad de cuánto puede deberse a vivir en ambientes polares o por relaciones filogenéticas, ¿no? Que yo espero que sean las dos cosas, pero ¿habéis pensado hacer mirar la señal?
Nosotros eso no lo trabajamos directamente, pero sí, desde luego tiene que ver también. En uno de los casos concretos que os he enseñado, una de las especies que tenía una responsividad del ciclo Basque muy fuerte en horas de noche, esa es una de las especies que biogeográficamente ha llegado más tarde a estas regiones, en este caso del Ártico. Entonces, una de nuestras hipótesis era esa, que le ha dado menos tiempo evolutivo, por así decirlo, a aclimatarse a esas condiciones en las que vive ahora. Pero desde luego, claro, lo interesante sería combinarlo con especialistas.
En invertebrados no salió algo así, ¿no? Como que había un poco de todo, pero sí que había una adaptación específica, no necesariamente por filogenia, y que conllevaba que un trade-off de adaptarse al frío, pérdida de tolerancia al calor, con lo cual estaba interesante. Bueno, una cosa para explorar. Y la otra preguntilla, Brachytecium, no sé si lo he entendido bien, seguramente mal, pero le has puesto como un caso llamativo y representativo. Claro, me pregunto si es porque Brachytecium vive en ambientes donde con mucha agua, pero a la vez que varía mucho el nivel de agua, entonces tiene ya una plasticidad natural de adaptación a esos cambios bruscos, ¿o por qué podría ser?
Sí, eso es algo de lo que no he hablado, pero las especies con las que trabajamos tienen, algunas de ellas, pues, distintos requerimientos microecológicos, y lo que vamos obteniendo hasta ahora precisamente es eso, tiene que ver con cómo de afines son estas especies al agua o no. Entonces, Brachytecium es una de las que hemos estudiado, que vive asociada estrechamente a cursos de agua dulce, pero que allí en Antártida muchos son efímeros. Y en este caso, en nuestra hipótesis, esta es una de las especies que peor está tolerando de momento la desecación y la congelación. Entonces, nuestra hipótesis es que está, digamos, aclimatado a vivir en un ambiente más amortiguado, porque es más frecuente para él que sí haya agua líquida durante ciertos periodos de tiempo. Y en ese agua, pues bueno, la temperatura también es un poco más estable.
Bueno, pues vamos a dar ya por finalizado el seminario. De todas maneras, como se suele hacer habitualmente, iremos al kiosco. Si alguien quiere hablar algo más con Beatriz, se puede hacer. Y bueno, simplemente agradeceros a todos los que estáis aquí, también a todos los que lo han seguido online, y también, como quedará en YouTube, pues también es posible que otras personas puedan verlo después. Entonces, nada, como he dicho al principio, este es el primero de 3 antes de Navidades, y el próximo viernes último de mes tendremos a Thomas Smith, que nos hará el seminario. Y al final de noviembre tenemos otro seminario más. Entonces, nada, gracias a todos y hasta el próximo.